Okay, good morning, and we're going to get started here on, we're going to wrap up on chapter 24. We're going to finish talking about electron transport, and then we're going to start on bones from the physiology side of bones as opposed to the anatomical side, what we've been doing in lab. You may have noticed I have pushed back the due date on the lecture exam to give you a little more time to get ready for it. And if it says, I may have to, I'll obviously have to change the dates on the third exam because those dates were premature to pick that. So don't worry about those dates. Anyway, anybody have any questions on anything? Okay, I'm going to go ahead and go into, here we go, metabolism. And since it was quite a while ago, since we only met one time last week, I'm going to start here on the discussion on ATP. Everybody should see the slide that says ATP, and I hope everybody can hear me. So do the best we can here. So ATP, this is what it's all about. Everything that we're leading up to is for the formation of ATP. Our cells use ATP as their money. They pay their bills. Cells pay their bills with ATP. We can't take the energy from glucose directly. That would be very bad. There's too much energy in a molecule of glucose for us to process at one time. So we have to do it slowly. We have to do it step by step. And we can use glycolysis, or we can use cellular respiration, well, electron glycolysis, or we can use glycolysis or electron transport as the ways to get our energy. The end result is still going to be the formation of ATP. Whenever we need to do something, contract a muscle cell, for example, or to generate a nerve impulse, or to pump the sodium-potassium pump, we need energy. So we grab a phosphate and we grab ADP, and we take energy from glucose and we jam the third phosphate onto ADP and make adenosine triphosphate. And that third phosphate is held in place with a temporary high energy bond. It only lasts 7 seconds before it breaks down. But for 7 seconds, we can send that ATP molecule anywhere we want in the body. Now, every cell is busy making ATP, so it isn't like we have to make one on our finger to send it to our toe, for example. But it only takes, it's not inconceivable because it only takes 20 seconds for a red cell to go through our entire body and come back to the heart. So, but we use ATP. We break the third phosphate bond, we release energy. When we pump the sodium-potassium pump, ATP reacts with the sodium-potassium pump, transfers its phosphate. It transfers its third phosphate to the sodium-potassium pump. Anytime we want to get energy to do something, we take the third phosphate off of the ATP with its energy, and that energy becomes available to do something. Then we reload it. The term phosphorylation is simply adding that third phosphate back on. And it takes energy to get that to stick. Now, and that's a good thing. We use energy there so we can take the energy slowly from glucose, put it into ATP, take it to the cell, break the bond where the third phosphate is attached, transfer the phosphate temporarily to the sodium-potassium pump or the skeletal muscle fiber or to the nerve cell, and make it work. Now, the ADP and the phosphate are not are not damaged. They are just put back together again. We have more energy, and as long as we have energy, we can take the inorganic phosphate, which is what we call the third phosphate, the inorganic phosphate. As long as we have energy, we can take that and jam it back on the ADP, make it work. And this is how we spend our energy. This is our energy budget uses this process of ADP and ATP, because this is what the ATP molecule looks like. It looks a lot like a nucleotide for RNA. It's got a nitrogenous base here in the left-hand side, the ribose sugar in the middle. It has nothing to do with RNA. I'll just tell you that it has nothing to do with RNA. It just looks like an RNA nucleotide. And over here are the 3 phosphates, and it'll go back and forth between its diphosphate and triphosphate state. As long as there's energy available to put that third phosphate on here, and we can park that energy in that bond for 7 seconds, then we can transfer that phosphate to some other compound to get something done. And this is what cellular respiration is all about. Glycolysis gives you a little bit of ATP, not a lot. Remember, glycolysis is done in the cytoplasm. It's done without oxygen. The end results of glycolysis are either the formation of pyruvic acid in the presence of oxygen or lactic acid if there is no oxygen. If we have oxygen available, glycolysis produces pyruvic acid, 2,3 carbon compounds. If we don't have oxygen available, we form lactic acid. We only form lactic acid when we've depleted the oxygen reserves in our muscle cells. Oxygen is stored in myoglobin, that's that red pigment. It's a respiratory pigment that we have that makes our muscles red. It's sort of like hemoglobin. It's called myoglobin because it's found in the muscles. This is why when you go to the store and you go down to the meat counter at Food City, for example, the beef is red. Beef is red because it has lots of myoglobin. You know, and when you get a piece of beef that you order a steak or something or a burger, and you say you want it rare, and it had and people say I want it blood rare. Well, that's not blood oozing out of your burger. That's myoglobin. The blood has long been drained from that meat. We don't want that blood around. All we have left is the myoglobin. And so the myoglobin is how our muscles store oxygen. And as long as we have stored oxygen, we're going to generate pyruvic acid from glycolysis. But when we deplete that oxygen, and we do that if we engage in long duration strenuous activity, like running a marathon. If we're going to run a marathon, and I know some people that do, I'm not one of them, but if we're going to run a marathon, you're going to end after running 26 miles plus the several hundred yards, you are going to have depleted all the oxygen reserves in your myoglobin, but your muscles are still going to function. Glycolysis doesn't need oxygen. It's not going to produce pyruvic acid though. It is going to produce some ATP for you. It won't produce pyruvic acid, it'll produce lactic acid. And that lactic acid buildup is what makes your muscles sore. If you engage in strenuous long duration activity and you deplete your oxygen, your muscles are going to be very sore for about a day. And it isn't so much of them being stretched, it is the fact that lactic acid accumulation is somewhat painful. It hurts, or it makes you sore a little bit, then it'll go away the next day. The oxygen debt will be paid back in about 24 hours. Your muscles can function just fine running anaerobically. They're still going to contract, they're still going to move because there's plenty of stored energy. Even though the oxygen's been depleted, the glucose is still available in the form of glycogen. So you can still see your muscles are still going to contract at the end of the marathon and while you're walking around later on. But let's assume that we've got oxygen. So we produce pyruvic acid. The pyruvic acid goes into the mitochondria. Now remember, everything up to this point, glycolysis takes place in the cytoplasm. And you only get 4 ATP from glycolysis. You only get, because remember, but you have to burn 2 ATP to make 4. So you're really only looking at a net gain of 2 ATP. Okay. So on a test question, read it carefully. If the question asks you total ATP from glycolysis, it's 4. If it asks you net, it's 2. Because you had to burn 2 to make 4. You had a net gain of only 2. So read the question carefully. I've noticed that a number of people across all the classes will see the question and not read it through and end up, answering what they think the question said. And I can see what they're doing. So. Okay, now, so we got the pyruvic acid going into the mitochondria. From there, those 2 3-carbon molecules, remember it's 2 3-carbon molecules because it split the glucose in half. It goes right into the matrix of the mitochondria, but as soon as it enters the mitochondria, it is going to be immediately oxidized, I guess is a good term. By an enzyme, a carboxylase enzyme, and it's going to strip off a carbon atom along with 2 oxygens. It's going to form CO2. So the first CO2 comes off as soon as the pyruvic acid enters the mitochondria. And this is all considered part of the Krebs cycle. So what do you need to remember about this? Well, carbon dioxide comes from the mitochondria. All of our carbon dioxide is generated from mitochondria. But we're only going to generate CO2 if we have oxygen available. We're only going to generate CO2 if we have oxygen available. If the cells are alive and the cells have oxygen, they're going to be generating carbon dioxide. Muscle cells are probably not going to generate a lot of carbon dioxide because they're not making pyruvic acid. All of our carbon dioxide comes from our mitochondria. So we've stripped off a carbon dioxide. We have a 2-carbon compound left. It is oxidized by NAD+ to grab 2 high energy electrons and 2 hydrogens. And the remaining, and what's left over reacts with coenzyme A to form a compound called acetyl-CoA, which combines with a 4-carbon compound from the very end of the Krebs cycle to form citric acid, which is why it's often called the citric acid cycle. Now, why is this important? Is anybody going to ever come up to you? In fact, I don't think anybody's ever asked me, walk up to me and said, hey, what happens to the acetyl-CoA? Where's it go? I don't think it's ever come up in conversation. I don't ever remember hearing anybody in healthcare asking someone else. I've never, my doctor doesn't, and I know my doctor likes to discuss things with me. He's never asked me about what happens to acetyl-CoA. So it's just a step to get us into the Krebs cycle. That's all it is. But what's more important from the Krebs cycle perspective is not the formation of acetyl-CoA. It's the fact that you're going to gain a lot of high energy electrons from the Krebs cycle. This is what the Krebs cycle does. the most important thing the Krebs cycle does is capture a whole lot of electrons and hydrogens. Because right now, they're more important than the handful of ATPs that we generate. Because remember, you only generated 4 ATP from glycolysis. The Krebs cycle is only going to give you 2 ATP. That's not very much. Remember that our glucose molecule has about 680 kilocalories of energy available. Each ATP only has 7.3 kilocalories of energy. So if you generate 6, that's something like 45 kilocalories of energy out of 680. That's not a very good return. That's not even 10%. That's like what, 8% if I'm doing the math right, which I probably aren't. Can't, won't, whatever. And that's not very good. That's only a little bit of energy. We can't depend on glycolysis to do everything. It'll keep us going. It'll meet our needs, at least temporarily. But that's not very much energy. I mean, 4 ATPs from glycolysis and 2 from the Krebs cycle, 45 kilocalories of energy is not very much. What's more important than the small amount of ATP that glycolysis and the Krebs cycle are making are the capture of high energy electrons. That's what the Krebs cycle does. The Krebs cycle will capture high energy electrons. And that's more important than anything else it does. And how it does it is by taking a 2-carbon compound and a 4-carbon compound and continuing to oxidize it with NAD plus, which is a temporary electron acceptor. It's not the ultimate electron acceptor. Remember, when something is oxidized, it gives off electrons and it releases energy and it releases hydrogens. So we use a temporary electron hydrogen acceptor like NAD plus, and it grabs those hydrogens and it grabs those electrons. And so we're going to oxidize this molecule, this 6-carbon molecule, the citric acid we form. We're going to oxidize that around the Krebs cycle, and we're going to strip off 2 more carbons, and we're going to, for every pyruvate, we're going to get rid of 3 CO2s. That's just what we have to do to get those high energy electrons. So that's more important than the ATPs, the high energy electrons and the hydrogens that go with it. Okay. So this is what happened. This is the first step in the Krebs cycle. The pyruvate, remember, you get 2 pyruvates from glycolysis. So there are 2 pyruvates going in one after another. They probably go in at the same time. I don't know. But we always say it goes in one after another. So pyruvate comes in. It immediately is decarboxylated, meaning we strip off the carbon and we take 2 oxygens with it, CO2, carbon dioxide coming off right there. The remaining 2-carbon compound is oxidized by NAD+. NAD+ is, it's an electron acceptor. It grabs 2 electrons, it grabs 2 hydrogens and goes off. Then we react it with coenzyme A and we get acetyl-CoA. Okay, that's great. Okay, we made what we needed to sort of prime the pump of the Krebs cycle. And we start the Krebs cycle with citric acid. Citric acid is just the combination of acetyl-CoA and oxaloacetate, a 2-carbon and a 4-carbon. So now if you want to get picky about this, for every pyruvic acid that goes into the Krebs cycle, I don't even like this. For every for every acetyl-CoA that goes into the Krebs cycle. No, forget this. This is going to confuse you. I'm going to just go on to this to the sum total. Okay. And if you get really bored someday, you can watch this animation here. Unfortunately, it doesn't stop it when you want it to, but this is what happens with the pyruvate. Each pyruvate will give you 3 CO2s. Each pyruvate will give you 4 NADHs. There's our first, there comes our CO2, our first NADH, our second NADH, another CO2, our third NADH, and 1 ATP, an FADH2, and another NADH. Now, what is so important about those NADHs? There's 2 little yellow dots there that represent the high energy electrons. You have 2 hydrogens and 2 high energy electrons in every one of those NADHs. They've been reduced. They've captured the electrons and the energy coming off of the compound that's being oxidized in the Krebs cycle. This is what the Krebs cycle looks like, more or less. We count what starts up here when pyruvate comes in. There's your first CO2 coming off, your second CO2 coming off, your third CO2 coming off. There is NAD plus being oxidized. There NAD plus is gaining electrons. It's becoming reduced. It has picked up electrons and hydrogens. There it is again and again and again. Now, when we exercise and we're burning glucose, we generate more and more CO2 as we do that. The Krebs cycle goes around faster and faster because the more glucose we burn, the more glucose we're going to oxidize, the more energy demands we have, we're going to release more CO2. The more CO2 you have, the faster you're going to breathe. Because the CO2 is going to, you don't, as I've said many times, you don't breathe faster to get more oxygen, you breathe faster to blow off the CO2. Because our brain doesn't monitor oxygen levels, our brain monitors CO2 levels. It's easier. We monitor CO2, we use that as an indicator when to breathe. The more CO2 we have in our bodies, the more carbonic acid it forms in our blood. That makes us more acidic. Our brain says when our pH drops, we need to breathe faster to get rid of the CO2. It's a very nice side benefit that as we're blowing off the CO2 that we're gaining more oxygen. So the Krebs cycle will go faster and faster, going around in its cycle faster and faster as we burn more glucose, as we meet the energy demands. And so it's a measure of how efficiently we burn glucose by how much CO2 we're producing. So it's sort of a relevant piece of information. It's relevant to know where the carbon dioxide is coming from in our bodies. It's coming from the cell's mitochondria. So what do you need to remember? Well, this is the bottom line slide right here. 8 total NADHs. This is from a molecule of glucose. Because remember, there's 2 pyruvates. So you have to add everything together. You have to add the results of the first pyruvate and the second pyruvate. And so you end up with 8 NADHs, 2 NADHs from the first, when the pyruvate comes in, and 6 more from the cycle itself. You get 2 FADHs. So from all this, you get 8 NADHs. That means you get a lot. You get each one of those molecules, the NADH molecule is containing 2 high energy electrons and 2 hydrogens, because they all have to stay balanced. Here we have FADH2, another compound that can be reduced, you know, a temporary electron carrier, if you will. And so we have even more high energy electrons down here, 2 in each one of those with 2 hydrogens. So this side over here, the NADH and the FADHs, that's more important than anything else the Krebs cycle does. That's what the Krebs cycle is known for, capturing high energy electrons and hydrogens. I can't emphasize that enough. The Krebs cycle is known for it. The most important thing the Krebs cycle does is capture high energy electrons and hydrogens. In doing so, it breaks apart bonds and it releases CO2. The only place our CO2 comes from is from the Krebs cycle. And we pick up 2 ATPs. Okay, so if you're keeping score, we have a grand total of 6 ATPs made between glycolysis and the Krebs cycle. If you count the net gain, it's only 4. So we haven't really done a whole lot. Remember, there's 680 kilocalories of energy in a molecule of glucose, and we've gotten 45. That's not gonna, it's not what we want. We want a lot more. Okay, so we move into the inner membrane of the mitochondria. Remember, the mitochondria has like this. There's the outer membrane of the mitochondria. These folds are called cristae, and these are folds of the inner membrane. And where the Krebs cycle takes place is in the matrix area in here in the middle, but the electron transport chain is embedded in the surface of the inner membrane. And there's a space between the inner and outer membrane called the inner membrane compartment or inner membrane space. Nobody thought very hard about naming it, but there's a space in here. And that becomes very important to us here in a second. Now let's move back here. So, okay. So in the electron transport chain, you've got 3 enzymes that function as pumps. They're embedded in a cluster. And we don't really need to remember the names of these enzymes, but their role is to oxidize the NADH, capture the high energy electrons, and pump the hydrogens that come across. Because when you oxidize NADH, you're going to break those bonds in there. You're going to release their electrons that they've captured. You're going to release those high energy electrons they've captured and release their hydrogens. Now, these enzymes embedded in the membrane are going to use those electrons and use their energy, their stored high energy, to pump hydrogens across the inner membrane space, across the inner membrane into the inner membrane space. So these little pink dots up here, those are hydrogens. Those hydrogens have been pumped across. See, because when you oxidize NADH, you strip off electrons and you strip off hydrogens. The electrons are used to pump the hydrogens across here. Okay. So this goes a little faster than I'd like it to, but here are the 3 enzyme complexes here, and you'll see an NADH come up here fairly quickly. And there it goes, and those are electrons. Those little things moving across are electrons. See, because we pass electrons from one complex to another. We use that energy to pump the little white dots of hydrogens. We pump the hydrogen across. So it is a step-by-step process. The NADHs and the FADHs are oxidized back to their low energy state. NADH goes back to being NAD+. FADH2 goes back to being FAD. The hydrogens and the electrons are taken off of them. The electrons are captured by these enzymes here, and they use that energy to pump the free hydrogens from one side of the membrane to the other. And they end up out here in the inner membrane space. And the electrons are going to get passed from one to another to another. So we split apart the hydrogens and the electrons. The electrons stay embedded in the electron transport chain, but they are passed, they are shuttled from enzyme 1 to enzyme 2 to enzyme 3. And as they do so, they release their energy, and that powers those enzymes. They function as pumps, and they pump the hydrogens across the membrane. This works a little slower here so you can see it. There's the chain, same phosphate lipid bilayer. Here comes NADH. It gets oxidized by this enzyme. Hydrogens that get released are pumped across the membrane into the inner membrane space. The electrons are transferred from this enzyme to this enzyme to this enzyme. We keep pumping hydrogens across the space. Now, eventually those electrons lose all their energy and drop back down into the matrix. In the meantime, all these hydrogens up here would like to achieve equilibrium with the hydrogens that are in the matrix. Because remember, you're splitting apart. You split apart the NADHs down here in the matrix, so you have lots of hydrogens down there. You have even more up here in the inner membrane space. They want to diffuse across back down to the matrix to reach equilibrium. That's just what they want to do. There's a high concentration on one side and a low concentration on the other side, and simple diffusion wants to take place. The only way for those hydrogens to come back into the matrix is through this last enzyme called the ATP synthase. It will let the hydrogens come through. And as it does, it causes this enzyme to rotate. Yeah, it actually rotates, it pivots. And when it does, it forces inorganic phosphate onto ADP and making ATP. Watch, here comes the high energy they're pumping the hydrogens across, and the hydrogens now will find a way to come back down through here, through the ATP synthase. Now, there they go. And that causes the synthase to rotate, and it provides the energy to force the third phosphate onto the ADP. And now you've made ATP. You made a lot of it. Okay, so this gives me a little chance to explain that a little better, I hope. Here comes the NADH carrying 2 electrons and 2 hydrogens. This is the stuff that we got from glucose. These are the electrons and hydrogens that were stripped off of glucose when we oxidized glucose. We started the whole process of oxidation back in the cytoplasm. Glycolysis, yeah, it makes some ATPs for us, but it also captured some, it made 2 NADHs with their high energy electrons and their hydrogens. And that was more important than the little bit of ATP it made for us. So here comes the NADHs from the foods that we've eaten. Everything energy-wise has been either glucose or converted to glucose. The NADH approaches the first enzyme. It gets oxidized. And when something is oxidized, it loses electrons, it loses energy, it loses hydrogens. Sure enough, the electrons and the hydrogens and the energy have come off of the NADH, and it's oxidized back to NAD+. The electrons go into the enzyme. the hydrogens get pumped across the membrane. They're actually split apart now, because normally hydrogens and electrons travel together. They're split apart. The hydrogens go up here. The electrons are used to power them, to power the pump to move them. The electrons are transferred to the second complex here. In the meantime, FADH2 comes along, it gets oxidized and gives up its electrons and hydrogens. And it goes back to being FAD. It goes back to the Krebs cycle. The electrons are transferred to the second complex. We continue to power the pumping of hydrogens across the membrane. The electrons still have energy and they are passed to the third complex, where we continue to pump hydrogens across. As we run out of energy in the electrons, they gradually drop down through the third enzyme complex, and it lets them go back into the matrix. Now in the matrix, the electrons reconnect with their hydrogens. Where did the hydrogens come from? Well, because remember, the hydrogen concentration is always high out here. They want to reach equilibrium with what's in the matrix, so they're going to pass through the ATP synthase. Those hydrogens have to, let me back up. The movement of the hydrogens generates our ATP. It causes the ATP synthase actually to rotate and pivot, and the mechanical energy of the rotation forces the inorganic phosphate onto ATP molecules, and we make about 36 ATP right here. Up to now, we've only made 6 ATP. Here we make 36 of the ATP. So we make a lot of ATP. We capture a lot of energy right here in all the ATPs we make. We got a problem. All those hydrogens have come through the synthase. If we reach equilibrium with the intermembrane space and the matrix for the hydrogens, there'll be no pressure for the hydrogens to come across. The synthase won't turn. If it doesn't turn, we don't make ATP. We can't allow the hydrogens to reach equilibrium. So fortunately, this is where oxygen comes into play. Oxygen, is so important to us. Oxygen is our final electron acceptor. The electrons that are coming down here through this third enzyme complex find an atom of oxygen. Atmospheric air splits in half. Atmospheric air, atmospheric air is O2. We split that in half, and it grabs 2 electrons because it wants 2 electrons. It wants to have 2 more electrons to fill up its outer shell. The only way it gets that is to grab 2 electrons who are dragging their hydrogens with them. So conveniently, if we grab 2 electrons and 2 hydrogens, we make a water molecule. In other words, what happens here is the electrons, the hydrogens that have come through here are reunited with their electrons at the base of the third complex. And those electrons and hydrogens are grabbed by the oxygen atoms that are now present when we're breathing, with normal, not cellular respiration, but normal respiration. And we make oxygen available here, and we split it down to its, we break the molecular oxygen into the individual atoms, and we use that to form water. And if you remember, the formula for cellular respiration is glucose, C6H12O6, plus 6 oxygens will yield 6 CO2s and 6 water molecules and the release of energy. And that's what we're getting here. We have to keep the hydrogens concentration in the matrix always less than what's up here in the intermembrane space. So we grab those hydrogens. with oxygen. We grab electrons, we grab hydrogens. The oxygen is the final electron acceptor. I can't say that enough times to stress it's important. Oxygen is the final electron acceptor. This is where oxygen works. You know, in every cell in our bodies, this is how oxygen works. It grabs those electrons, grabs those free hydrogens, makes water. Keeps the hydrogen concentration lower than what's in the intermembrane space so that they will continue to come down through the ATP synthase and make ATP. If there is no oxygen available, and this is what happens when cells die. Cells die. If there is no oxygen available, then the hydrogen concentrations rise. We reach equilibrium. The ATP synthase doesn't turn, and we don't make ATP. We don't have energy. So when the cell dies, it's because there's no oxygen present. That's why oxygen is sort of important to us. There are toxins, gases, and poisons that will interfere with this process. It will block the oxygen from accepting the high energy electrons, the low energy electrons and the hydrogens. Cyanide, for example, classic poison, works by blocking the ability of oxygen to accept the electrons. So, and the cell dies. And and if it's sufficient there, your patient dies. So this is why oxygen is so important to us. So this is the electron transport chain here. Here go our hydrogens. They come back through the ATP synthase. Here comes an oxygen atom and combines with the hydrogens. All these hydrogens have come through. We got to get rid of them, and so we got to form water. The easiest way to get rid of those hydrogens is to form water with the available oxygen that grabs the extra electrons too. All those hydrogens come through there, and we form our water molecule every time. Now, every living organism does this the exact same way. Everybody does cellular respiration the exact same way. The whole mechanism works in, if you are an oxygen breathing organism, and there's a few out there that aren't, there are a few anaerobic type bacteria that function without oxygen. But if everything, 99.9999% of all living organisms do this. Everybody does this. Plants, animal, funguses, nearly all bacteria, anything. Viruses don't care. Viruses aren't alive, but everything else does. So how much ATP do we get? Well, different books say different things. Some say 32, some say 28, some say 34. Generally take it as an average number, about 36 ATP. That's a lot. That's about 260 kilocalories, somewhere 260, 280 kilocalories a minute. That's a lot of energy. That's 40% of the available energy found in a molecule of glucose. That's really good. Granted, 60% of the energy is lost as body heat. That's okay. We're designed to run at that temperature, at normal body temperature. 40% is available to do stuff. Muscle contractions, nerve impulses, sodium potassium pumps, anything that requires energy, any type of active transport that requires energy will get its energy from ATP. And we make our ATP, nearly all of our ATP gets made through electron transport. Now, the synthase does its job very well. It actually rotates when the hydrogens come through, and that rotation causes the ATPs to get made as the inorganic phosphates and the ADPs are combined. And in some cases, when the cell runs out of oxygen, and it's threatening to reach temporarily and push hydrogens back into the intermembrane space so they can come back through. It can't do that forever. Forever is a very short term in cell time. It can do it for a few minutes. And so when the cell runs out of oxygen, it will, in its last effort to stay alive, it will pump hydrogens back through the synthase so they can come back through again and make some more ATP, even though there's no oxygen there to grab those electrons and hydrogens. So this is a little more complicated diagram of it. It's fascinating to think that this was actually determined. We're talking something that is, embedded in the cell membrane. So we know how small the cell membrane is. But this is how it works. The hydrogens come through and cause the rotor to spin. And when the rotor spins, it causes this knob to spin here. And the movement of the knob is what forces the inorganic phosphates onto the ADP. The mechanical energy gets transferred. We're taking chemical energy and transferring it to mechanical energy back to chemical energy. So if you want to see the whole picture right here, you can. This is one of those sources that says 28 ATP, but again, every textbook's going to be a little bit different. We tend to sort of focus on about 36 ATP. You won't be wrong if you select 36 ATP as an answer. You won't be wrong about that. Because some, it just depends on how this is added up. So anyway, a couple terms to know. Glycogenesis, the creation of glycogen. Glycogenesis. When we have more glucose than our cells can use, we start parking it in glycogen. Glycogen is our body's polysaccharide. We find this in our liver. We find it in our skeletal muscle. It's what keeps the skeletal muscle cells alive. Lets them run, lets them function anaerobically when oxygen gets depleted. This is why skeletal muscle cells take a long time to die. Skeletal muscle cells will often live for hours after death. It takes 8, 10, 12 hours for all the skeletal muscle cells to die. Cardiac muscle cells take an hour to die. So these cells, we'll talk all about that when we get into muscles very shortly. But skeletal muscle cells take a long time to deplete their stored oxygen and their stored glucose. Now, glycogenolysis, the breakdown of glucose. We take stored glycogen and we break it down into glucose. We use our lysosomes to break apart the glycogen to get glucose. And so that's how we use that stored energy. And then we have my favorite term, gluconeogenesis. Gluconeogenesis is the formation of glucose from lipids and proteins. When our energy levels become depleted, when our carbohydrate levels have been broken down to practically, when we've exhausted our carbohydrate levels, we can start breaking down fats and proteins and converting them to glucose. Gluconeogenesis. This is what happens. This is why we can go for about a month without any food at all. This is what you see frequently in your pregnant female. We'll talk about this. In fact, I just talked about this in my AP2 class. Your pregnant female will be to make sure that the baby gets as much glucose as possible, because the mother provides the glucose and nutrients for the baby. There's no exchange of blood, but there's exchange of nutrients and waste and gases. Anyway, the mother will undergo gluconeogenesis, breaking down her fats and breaking down her protein for her glucose needs, so the glucose that she obtains from her diet goes straight to the baby. So, so these are three terms that you're going to hear a lot of, particularly gluconeogenesis. So, and that should be, okay, that's the end of chapter 24. Now, and we have enough time to start chapter, what would be called chapter 6, the bone chapter. So the test on chapter 3 and 24 doesn't, you know, the content on the test stops here. I think I've pushed it back a few more days. I'll have to go back and check, but I was pretty certain I slid this test back to the 29th. We're going to go on and look at new material, but I wanted, you know, with the bone test and this running in the bone test in lab and this test coming up, I wanted to make sure you had plenty of time to get everything done. In the meantime, we're going to go ahead and start new content. So let's see if we've got that up already. Okay. Yes, let's get out of here. Back to okay. If I did this right, you should see a slide that says Chapter 6, Bone Tissue and the Skeletal System. Now, I will tell you that from here on out, this class is going to get a whole lot more interesting because we're going to start dealing with organ systems and the things that go wrong with them. Up to now, I've had to go through all the content to get us here, you know, cells and cell structure and the chemistry involved and where the energy comes from and how we make proteins. And the worst is over. From here on out, we start talking about organs and organ systems. So we start with bones. We spent a lot of time looking at bones in lab. Now we're going to look at how bones function. And this is where you start to enjoy this part of this class. So, because up to now, I know it's probably been pretty tedious. So our skeleton is bone, but it didn't start that way. It started out in utero when we were developing baby in utero. We had a full skeleton in place 6 weeks after conception. Now we're an inch long at that point. So we're an inch long, and yet we have a complete skeleton available to us. And that skeleton was made out of hyaline cartilage. By the time we're about 23 or 24 years old, we have finally replaced all of that hyaline cartilage. We don't close all of the gaps in our bones. They're not really gaps, they're locations where hyaline cartilage is, till we're about 23 years old, maybe 24. Up until that point, we are in the process of undergoing what we call ossification, the conversion of cartilage to bone. Now, a lot of it takes place in utero. So we have a lot of skeleton in place in utero, but we have a lot more that's still cartilage that takes another 20-some years to get through. And unfortunately, this is not how our bones work for muscles. So anyway, bones do a lot of things for us. They support our body. They hold the soft organs in place. They protect the soft organs. The viscera is protected in the thoracic cavity. The brain and spinal cord are protected by the skull and the vertebral column. We can't move as well as we can just by using our muscles. We have to amplify their strength. Muscles are attached to bones so that so our bones function as levers. We're going to see that starting in lab today. So our muscles pull on bones to make our strength greater because they're not strong enough. Muscles are not powerful enough to get us to do the things we want to do. So we amplify their strength. Our bones store calcium and phosphorus for us. That's where 99% of all that is found in our bodies. It's a big reserve for those two very important minerals. We make blood cells in our bone, the term hematopoiesis. We make the red cells, we make all 5 white cells, we make our platelets in bone. We store fat in the middle of our long bones, and we produce hormones in our bones. There is a hormone called osteocalcin that regulates our insulin levels. It regulates our glucose levels and helps to control our metabolism. It isn't just, we don't just depend on insulin to control our blood sugar. We use cortisol from our adrenal glands, and we use osteocalcin from our bones. So a lot of things we need our bones for. Bones are also living tissue. Bones are living tissue, something we all tend to forget. We see a skeleton, you know, it's not alive, right? But in our bodies, it is. There are living cells embedded in the matrix of the bone. Millions of living cells linked together. The osteocytes are living cells, and they can build the bone up, they can tear the bone down, they can do whatever they want to. They respond to stress and movement, and they can cause the bone to grow or to shrink. So bone is living tissue. Now, our bones, as you know from lab, can be flat, like the sternum or the scapula or our ribs. They can be irregular, like the vertebrae. They can be long bones. I love this definition. Long bones are longer than they are wide. So one of your finger bones, one of your phalanges, and your femur, one of the shortest bones in our bodies and one of the longest bones and the longest bone in our body, are both long bones. Doesn't matter, as long as they're longer than they are wide, they're a long bone. And then we have the short bones, the carpal bones in our wrist, the tarsal bones in our ankle. The sesamoid bones, like our patella, is a short bone. You know, shaped like a sesame seed. That's why they're called sesamoids. And this is the various shapes. You're all familiar with these from lab. Now, bones are considered an organ because they're not just a mass of calcium. There are a variety of bone cells. There are bone builder cells called osteoblasts. There are bone destroying cells called osteoclasts. There are osteocytes, the living bone cells, and there are bone stem cells that can make all forms, all the other 3 types of bone cells. Bone also contains blood vessels and connective tissue and nervous tissue and cartilage, and muscle cells are attached to it, and epithelial cells line blood vessels. So bones are considered organs because they have multiple tissue types in here. If we look at our bones, we can look at it in 3 different levels: gross anatomy, microscopic anatomy, and chemical makeup. Gross anatomy wise, there are 2 types of bone: compact bone and spongy bone. A compact bone is the hard bone that you see in the outer surface of any bone that you look at, whether it is whether you're looking at a femur or a fibula or a vertebrae. The hard bone on the outer surface, the hard structure on the outer surface of that bone is called compact bone. It makes it look smooth, it makes it look solid. It's where muscles attach. Muscles will always attach to the compact bone. It's very strong. Underneath that bone underneath the compact bone is what's called spongy bone. Spongy bone looks like a sponge, looks like a honeycomb, or a sponge. It has a series of structures called trabeculae that look like little cross bridges that go that you see with like A sponge is a bunch of little holes with, the little tiny hole. You know what a sponge looks like. And this is what the spongy bone looks like. Now, the advantage of spongy bone is that it's very, very strong. It doesn't look like it'd be very strong, but it is very, very strong. We have a lot of spongy bone in areas where we're going to be stressed from different directions, because those trabeculae can take the stress and spread it out so that no one structure, no one portion of that bone is going to be under undue stress. Like, for example, here is the head and neck of our femur right here. Here's the compact bone running down the shaft. We need lots of compact bone in the shafts of our long bones because they're load bearing and they do a lot of work for us. But here we have the stress, where we have the stress of motion, where we're moving in all different directions. We have a thin coat of compact bone, and the head and the neck of the femur here, just like at the humerus, and anywhere where we're going to be exposed to stress, we're going to have this arrangement. The spongy bone here, and all these little lines are the trabeculae, will take the shock of motion and spread it out. What happens in osteoporosis is this is all dissolved away. And all you have left is just a thin outer shell of compact bone on top of the spongy bone. Inside of this, you have fat, what we call the yellow marrow or red marrow. Red marrow is the blood cell forming tissue. Yellow marrow is fat. Okay, so that's what we deal with compact bone and spongy bone on our long bones. Now, the term diploe is how we describe spongy bone. Diploe is a bone sandwich. We find this in our flat bones. We find this in the irregular bones. This bone sandwich has a thin layer of compact bone on the top surface of the bone and the bottom surface of the bone, with the spongy bone in the middle. Our ribs are a bone sandwich. Their outer surfaces on both sides are made of compact bone, and in the middle is the spongy bone. It makes the rib very strong, but also very lightweight. Spongy bone is a lot lighter than compact bone. A rib weighs a whole lot less than a femur does. Femur is heavy and bulky because it has a lot of compact bone in there. So we have this diploe sandwich in here, giving us a lot of strength without a lot of weight. Now, the outer covering of, and I want to come back to this a couple times, the outer covering of bone is called the periosteum. It's a layer of connective tissue that surrounds the outside. It's what holds the blood vessels and nerves attached to the bones that have to penetrate the various foramen to go into the middle of the bone itself. The inside layer of the compact bone, the lining of the compact bone on its internal surface, is called the endosteum. And from both the periosteum and the endosteum, we can grow new bone. So this is what the sandwich looks like. This is the diploe. Look at consider our skull. This is the parietal bone in the skull, right here. A thin layer of compact bone, another layer of compact bone with our spongy bone in here. These are the trabeculae. The spongy bone looks like a sponge, sort of what this looks like. See, the skull is very lightweight. It's very strong. It's very, very strong. These trabeculae take shock. Some sort of, any kind of shock that hits the skull, and they spread it out all over the place. So we don't, You bump your head and nothing happens other than it's temporarily painful. You bump your head and it hurts, but that's because you're hitting the skin more than anything. The shock there is no shock to your brain because the shock is transferred all around through these trabeculates, spread out. Doesn't bother us. So but imagine if our skull was all compact bone. Imagine how heavy it would be. Imagine how much more powerful the muscles of our neck and shoulders would have to be to hold our head up. We probably wouldn't like that. Of course, we probably wouldn't know anything any different because of that. We just had our necks would be much more massive to hold our head up because we'd be choosing to protect our brain with lots of compact bone instead of this a little more elegant approach here. We don't need a lot of weight for strength. This whole principle is applied in construction all the time. A concrete block, if you've ever looked at a concrete block, it has 2 holes in it. Concrete blocks used to be made a long time ago as a solid block of concrete. Bricks were made as a solid brick. You can still get bricks that are solid today. Bricks now have, I think, 6 holes or 6 or 8 holes bored through them in the mold, so that the brick, it has the outer wall of the brick intact. But if you look at it on the top of it, you can see these holes through it. That doesn't weaken the brick. The 2 big holes in the concrete block don't weaken the concrete block. They are just as strong using the thin shell for construction as they are as one that would be solid. The same applies to bricks. The construction industry took a page from the human body and said, hey, if the skull is just as strong with this spongy layer, and we're depending on the outer layer more and protect it here, let's do this. So we don't have to have solid bone, real thick solid bone to protect us. So we have this diploid. And this is a good picture of what diploid looks like. A layer of compact bone, another layer of compact bone, and a layer of spongy bone in the middle here, giving you a great deal of strength without a lot of weight. So that's the structure of our bones. We either have compact bone with a little spongy bone, or we have a lot of spongy bone and a little compact bone. If you're looking at a long bone, you have a couple terms here. The shaft of the bone is called the diaphysis. So the shaft is called the diaphysis. The ends of the bones are called the epiphyses. And the epiphyses are where growth is going to take place, at least up until we're about 23 or 24 years old. So the diaphysis is the shaft. The epiphyses are the ends of the bones. It's also, as I said, it's where bone growth takes place. There is usually a line on the bone called the epiphyseal line. That is the remnant of where the growth plates were located. The growth plates were called the epiphyseal plates. And those plates are where hyaline cartilage still existed. Now, up until age 23, we had growth plates in all of our long bones. The growth plates were hyaline cartilage. They would grow and they would be converted into bone through calcification. And then once we stopped growing, once we finally reached our adult stature, then those growth plates, the epiphyseal plates, would fuse, and all we'd have left is a white line. And so you can see here is a typical long bone. There is the epiphyseal line right there. Now, if you look at an X-ray and you see a white line on the epiphyses, it tells you that your patient is probably over 20 years old. If you see a dark line, it tells you that they're under 20, 23 years old. So if you are in a position where you can't get any information from your patient and they have a fracture and the X-ray shows a dark line, it tells you that their age is probably under 23. If there's a white line, it says their age is over 23. You have no other information there. It's just a simple process. It'll give you a sort of a ballpark figure to work with. So, but this pretty much holds true for everybody, that the epiphyseal line shows up after the growth plates have closed, and they don't close till we're about 23 years old. So it's sort of an approximation. It sounds better than ballpark. Anyway, this is your typical long bone. This is the epiphysis at one end, and the other end here. This is the diaphysis, the shaft. The cavity in the middle contains, usually for most of our bones at our age now, it's going to contain yellow bone marrow, which is fat. At our age, as adults, we only have a very few places in our bodies now where we have red bone marrow. Red bone marrow is what makes blood cells for us. There's only a few places left in our bodies that make blood cells for us. But when we were children, most of our bone marrow contained blood-forming cells. So this is what the plate looks like. Here we see an epiphyseal plate, a growth plate. This is hyaline cartilage in here. This dark band, is soft tissue. It's hyaline cartilage. Soft tissue doesn't show up on X-ray. So, it's solid, but it doesn't. It is. Stop that. Why did it do that? Hang on a second. There we go. Don't know why it did that. Okay. Soft tissue doesn't show up in X-ray. What we get is a dark band where soft tissue is located. Here we see the epiphyseal plate on the distal end of the femur and on the proximal end of the tibia, and also on the fibula. So the fibula is actually contributing something here. Once we get past 23 years, it generally turns into just a white line. And we can see that this patient's probably getting pretty close to it, because we see more white line present here than we see dark spaces. Now here in the tibia, so we're not done growing yet in this patient, because this is a pretty well-defined dark line here. This is the epiphyseal plate in here. Eventually, it'll just be seen as a white line on X-ray when it closes. So looking at this, your patient here is probably 21, 22 years of age, because it's almost closed, but not yet. So, and here we see what the adult looks like. This is the epiphyseal line right there. All we see is the scar from where the growth plate was here on the proximal end and the distal end. Here we see it. This is, as a child, this is a child's femur and tibia and fibula, and the plate is quite pronounced here. A continuous dark band across here, because that's where the hyaline cartilage is located. But once that's gone, all we get left is this scar, the epiphyseal line. The plate versus the scar. Long bones typically have a lot of spongy bone underneath compact bone at their ends in the epiphyses. So that's where you're going to see the greatest amount of stress. Here we see a typical long bone up close. We see compact bone on the outside. We see spongy bone as we approach the medullary cavity. That's where the fat, the yellow marrow, the bone marrow is located, or the red marrow is located. We see a thick layer of compact bone here, particularly on the shaft. Our periosteum is lining the outside of our bone, and there's the endosteum lining the inside layer of the compact bone. And then we have the spongy bone present here. So a little more about the periosteum and the endosteum. The periosteum is a connective tissue membrane. It has two layers. It adheres to the surface of the bone on the outside. The outer layer, the fibrous layer, is sort of like Velcro. It adheres to the surface of the bone. It has a series of fibers that attach it to the bone. That's why I say it's sort of like Velcro. It's very hard to remove. If you've ever eaten ribs and you're gnawing on your rib and you just got that little thin tissue layer, the little membrane on the surface of the bone, you're just sort of chewing on that. That's the periosteum. It's fine. But that's what holds blood vessels and nerves to the bone. The Sharpie's fibers hold the periosteum to the bone, and the periosteum is holding the blood vessels that go into and out of the bone, along with the nerve, in place. Underneath this fibrous layer, the Velcro layer, is what's known as the osteogenic layer. Now, anytime you see genic, it means creation, it means growth. This is the bone growth layer. This is where we have stem cells. Osteogenic cells are bone stem cells. Anytime you see the term genic or genesis involved here, it means the creation. In this case, it's bone creating cells. The osteogenic stem cells will give us all of the other bone cells we need, including more osteogenic stem cells. So that's what stem cells do. Stem cells give rise to all the other cells we need. For example, in blood cells, we have one stem cell, one blood stem cell that will give us all 5 white cells, the red cells, and the platelets. In bone, we have one bone stem cell, the osteogenic stem cell, that will give us osteoclasts, osteoblasts. It will give us more osteogenic stem cells in here. So it will give rise to all the bone cells we need. On the surface of the bone, we have the osteogenic layer on the true surface of the bone. On the outer layer, we have the fibrous layer. The fibrous layer in the periosteum is on the outside. The osteogenic layer is underneath. And these layers will hold the blood vessels and nerves that are going to pass through what we call the nutrient foramens, the openings in the bone, because bone is not a completely solid fused surface. There are lots of openings. You saw that in the skull. Foramens are everywhere. We have lots of foramens. Couldn't see them on our models on the pictures here, but we have lots of foramens in our bones to allow blood vessels and nerves to go into and out of our bones. This is what it looks like. Here we have the periosteum right here. These are the Sharpie's fibers here that stick to the bone. Here is our nutrient foramen right there going through the periosteum into the bone itself. There's our compact bone here, and this is our yellow bone marrow, and the inner layer is called the endosteum. And we don't really see the osteogenic cells here. These are the membranes that we do see. The endosteum lines the inner layer of the compact bone, It lines all of the trabeculae of the spongy bone in here. It lines all the canals that pass through the bone. It also contains osteogenic stem cells. So the endosteum is similar to the periosteum. The endosteum is called that because it's internal, and so it sort of acts like, it has an action similar to epithelial cells. It lines all the openings. That's what epithelial cells do in our bodies. These are not epithelial cells. They act like them as far as lining all the openings, but the endosteum lines the inside of the compact bone. It lines all the trabeculae of the spongy bone. It covers every canal and channel and opening, and it also contains osteogenic cells. Stem cells are present there. And this is probably a good place to stop. We will pick up with this on Thursday. So let me get us out of here. And, anybody have any questions on anything before we go on? Before I get us out of here? Okay, if not, in lab today, we'll be starting to look at the skeletal muscles. So I'll see some of you then.